Don't let this be the last video of ours you watch. Subscribe and stay for what's next. The result came back 3 times. Same machine, same sample, same technician recalibrating the equipment before running it a 4th time, just to be certain nothing had gone wrong upstream in the pipeline. The expectation, going in, was small. A stray 2 or 3% of leftover code, the kind every genome carries. Filler nobody had bothered to name. What came back instead was 98%. 98% of the human genetic sequence doing something that no one in that room could explain, cite, or fully name, even after checking the calibration a 4th and then a 5th time. Nobody celebrated. One of the researchers involved would later describe the feeling not as excitement, but as a kind of quiet unease. The specific discomfort of watching a number refuse to behave the way a number is supposed to behave in a well-run laboratory. Machines are built to confirm what is already suspected, to return numbers that fit neatly into an existing model. This one kept returning an answer nobody had asked for, and kept returning it no matter how many times the question was reframed. This is the story of that number, and of what has happened in the decades since it first appeared on a screen. The theories built to explain it, the ones that quietly collapsed under their own weight, and the small, stubborn fragment of the human genome that has resisted every serious attempt to fully translate it into something ordinary. For most of the last century, the story taught in classrooms was clean. Genes were treated as the meaningful part of a genome, precise instructions, letter by letter, for building the physical machinery of a body, proteins, enzymes, the working structure of a cell. Everything outside those genes, the overwhelming majority of the sequence, was labeled junk, leftover material, evolutionary packing foam, accumulated across hundreds of millions of years, doing nothing, going nowhere, worth nothing beyond a name that made it easy for an entire field to stop asking questions about it. For decades, that explanation held. It was simple. It was teachable in a single class period. And it was, as far as anyone could prove with the instruments available at the time, entirely reasonable. Then came the Human Genome Project, completed in 2003, and a more ambitious follow-up effort called ENCODE. Researchers stopped simply locating genes and started asking systematically what the rest of the sequence, the so-called junk, was actually doing while nobody was looking closely enough to notice. What they found complicated the tidy classroom story considerably. Regions once dismissed as filler turned out to function like switches, flipping other genes on and off at precise developmental moments, sometimes years apart, sometimes seconds apart. Other stretches scaffolded the physical architecture of the chromosome itself, holding structure the way scaffolding holds up a building under construction long after the original blueprint has been forgotten. By the 2010s, the phrase junk DNA had quietly disappeared from most serious research papers, replaced with something closer to functional dark matter, a term meant to signal that this material is present everywhere, doing something, but cataloged nowhere near completely. The ENCODE consortium alone eventually involved more than 400 scientists across dozens of institutions, spending years cross-referencing chemical markers, RNA transcripts, and protein-binding sites across the entire length of the genome, one small region at a time. It was, by any measure, an enormous and serious undertaking, not a shortcut and not a guess. It sounded, to most audiences, like resolution. It was not. Even scientists working inside the ENCODE project disagreed with each other, sometimes sharply and sometimes publicly, about how much of that newly discovered function was genuine biology versus a statistic overstated for the sake of a compelling headline. Researchers are careful, even now, more than a decade later, to frame that internal debate as unresolved rather than settled. The consensus story had a crack running straight through its foundation before the story had even finished being told to the public that funded it. Here is where it gets complicated. The first hints of the problem showed up quietly, in the margins of unrelated papers, long before anyone framed it as its own field of study. Researchers comparing genomes across species kept running into the same small, stubborn category of sequence that simply would not line up with anything else on record. Present in humans, absent everywhere nearby on the evolutionary tree, too short and too specific to dismiss as noise, too consistent across individuals to dismiss as an error. Eventually, that category earned its own name. Buried inside that vast, unmapped majority of the genome are sequences researchers call orphan genes, genetic material with no ancestor. Not a distant one, not a modified or heavily mutated one, none at all, in any species ever sequenced. Most genes can be traced backward through evolutionary time, the way a name can be traced backward through a family tree, appearing in earlier, slightly different forms across related species. Generation folding quietly into generation, the resemblance fading but never fully disappearing. A haplogroup, for comparison, works something like a surname passed down not on paper but inside the body itself. A genetic signature inherited quietly across centuries without a single document ever being signed. Orphan genes have no equivalent lineage, no surname, no family tree to trace backward, no matter how far the search extends. They appear to emerge directly out of non-coding sequence, the part of the genome once dismissed outright as junk. With no intermediate ancestor and no relative in any related species, human or otherwise, living or extinct. One of the clearest documented examples is a gene called ARH-GAP11B. It was studied in a 2015 paper published in the journal Science, led by researchers at the Max Planck Institute of Molecular Cell Biology and Genetics in Dresden, working alongside Svante Pääbo's team at the Max Planck Institute for Evolutionary Anthropology in Leipzig. The gene exists only in humans and, later data would go on to reveal, in Neanderthals and Denisovans, but not in chimpanzees, our closest living relatives. It appears to have arisen from a partial, malformed duplication of an older, ordinary gene, sometime after the human lineage split away from other primates. A copying error that, by every normal expectation, should have simply broken and vanished. It did not vanish. When researchers introduced it into the developing brains of mice, and later ferrets and marmosets, it triggered the production of significantly more neural progenitor cells than those animals would ever produce on their own. Enough, in some cases, to force ordinarily smooth animal brains to begin folding, the same wrinkling pattern that gives the human neocortex its density, its surface area, and much of its raw cognitive capacity. A single gene, no ancestor, tied, plausibly, to the very thing that makes a human brain, rather than something closer to a chimpanzee's. Not by years, by something closer to a rupture in the record itself. Since that 2015 finding, researchers have identified additional human-specific genes following a similar pattern, sequences unique to our species, absent in close relatives, each one a small variation on the same unanswered question. None has been studied as thoroughly as ARH-GAP11B. All of them raise the same problem in slightly different words. The first theory says this is simply evolution behaving the way evolution has always behaved. Messily, opportunistically, without a plan, a blueprint, or a designer checking the work. Roughly 45% of the entire human genome is made up of transposable elements, mobile fragments of code that copy and paste themselves around the genome across millions of years, the way a chain letter photocopies itself endlessly through a population without ever needing anyone's permission or oversight. Mutation, duplication, and the accidental co-opting of these mobile fragments are well-documented mechanisms, observed directly in laboratories under controlled conditions, not merely inferred from a distance. New genes can and do emerge from raw, non-coding material through exactly this process. The mechanism is real, it is broadly accepted across the field, and in the overwhelming majority of documented cases, it is entirely sufficient to explain what researchers find. This explains that new genes can appear from material that was never a gene to begin with, given enough time and enough copying errors. It does not fully explain why several of the most consequential ones, ARH-GAP11B among them, appear to emerge so abruptly, with so few intermediate evolutionary steps, concentrated so specifically along the human lineage, rather than scattered evenly across the broader primate family tree, the way ordinary mutation would predict. The mechanism holds up under scrutiny. The timing is the part that resists a clean, satisfying answer. Researchers who studied de novo gene birth are careful to frame their own findings in probabilistic terms rather than certainties. The process is documented as possible, even likely in isolated cases. But the specific sequence of accidents required to produce a functioning, brain-altering gene in such a short evolutionary window has not been fully reconstructed step by step. The outcome is visible. The exact path that produced it in this particular case is not. The second theory looks outward instead of inward, towards something that arrived rather than something that simply mutated in place. Endogenous retroviruses, the fossilized remains of ancient viral infections that embedded themselves permanently into the genomes of our distant ancestors, make up roughly 8% of human DNA, a proportion 4 times larger than the genome's own protein-coding genes. Over many millions of years, more than 100,000 of these viral insertion events accumulated silently in the germline, passed down like an uninvited inheritance that nobody chose and nobody has ever been able to return. A retrovirus, in plain terms, works something like a letter slipped permanently into a filing cabinet by a stranger who once held a key. The stranger is long gone. The door was resealed generations ago. But the letter stays, reshuffled and recopied faithfully with every new generation that inherits the cabinet. Researchers are careful to frame this as one plausible contributor among several, not a confirmed origin story for the sequences in question. The data doesn't prove viral material built these particular orphan genes. It only fails to rule the possibility out entirely. Researchers studying these insertions have gone as far as building genome-wide maps cataloging thousands of individual proviral elements, tracking where each one sits, how old it appears to be, and whether it still shows any trace of activity at all. Most are silent now, mutated into inert fragments over millions of years, the genetic equivalent of scar tissue. A small handful are not. One, remarkably, was eventually co-opted by the body itself to help build the human placenta, a virus that infected an ancestor millions of years ago, repurposed by evolution into something the body now depends on to reproduce. This is the part the records don't explain. Viral insertion accounts for a considerable amount of foreign-looking material scattered across the human genome, material that clearly carries the structural fingerprints of an ancient infection. It does not account for the orphan sequences that carry no viral signature whatsoever, no retroviral fingerprint, no ancestral echo, nothing that traces back to any known source, biological or viral, terrestrial or otherwise. They simply appear, fully formed, with no visible point of entry, viral or evolutionary, that any current tool can locate. The third theory is the one most researchers are hesitant to say out loud, and for defensible, professional reasons. In 1973, Francis Crick, the same scientist who helped uncover the physical double helix structure of DNA 2 decades earlier, published a paper with chemist Leslie Orgel in the journal Icarus, proposing what they called directed panspermia, the idea that life, or at minimum the genetic material capable of seeding it, might have originated somewhere else entirely and been deliberately transmitted here by an intelligence that predated us. At the time, it was treated as serious, if highly unorthodox, scientific reasoning, not folklore, not fringe entertainment dressed up in academic language, but a real hypothesis, published by 2 credentialed, respected scientists, built partly on the strange, near-universal uniformity of the genetic code across every known form of life ever sequenced on Earth. Mainstream biology has largely moved past the deliberate transmission version of that idea. Almost no working geneticist today argues seriously for an engineered origin, and most would call the strong version of the theory unsupported. What remains genuinely unresolved is narrower and considerably stranger. Not who might have authored the orphan sequences, but why sequences with no traceable origin still exist inside the human genome at all. Decades after the tools capable of tracing that origin were built, refined, and pointed directly, repeatedly, at the problem. The gap itself is real and documented. The extraterrestrial explanation for that gap is not evidence of anything. It is simply the oldest unanswered question in the field, standing quietly next to a newer one, refusing to leave the room even after the lights have been turned off. Picture a sequencing lab well past midnight, the only light coming from monitor screens and the dim blue glow of a refrigeration unit humming somewhere against the far wall, the kind of sound that becomes invisible after enough hours on shift. The building sits empty except for one technician and the low mechanical drone of servers processing a sample pulled from an ordinary cheek swab hours earlier. Nothing unusual about the donor, nothing unusual about the request that generated it. The air carries the faint, sharp smell of ethanol reagent, the same smell that has clung to laboratories like this one for decades, soaked permanently into the walls the way old smoke clings to a bar long after the last cigarette. On screen, a program flags a fragment of sequence and returns no match. Not a partial match, not a distant, loosely related one worth a footnote. Nothing. The technician runs the query again, assuming an error somewhere upstream in the pipeline, some contamination, some rounding mistake in the reference database. The result does not change. Somewhere inside an unremarkable strand of a total stranger's DNA sits a piece of code with no known relative in any database on Earth. And for a moment, before the explanations start piling up, before the theories get assembled and argued over, and quietly set aside for the next shift, there is only the plain, uncomfortable fact of not knowing. Outside, the city is asleep. Inside, a single unanswered question sits quietly on a screen, waiting for a morning shift that will not have an answer either. Here is where the story stops being simple. The twist is not that something was added from outside, smuggled in by a hand that was never human. The twist is that the material once thrown away as evolutionary debris turned out to include some of the exact sequences most directly responsible for what makes a human brain distinct from every other brain on the planet, including the brains of our closest living relatives. The very code dismissed for decades as garbage, filler, static, nothing worth the funding required to study it properly, is now understood to sit near the center of human cognition itself, close to the machinery that makes reasoning, language, and self-reflection possible in the first place. Wieland Huttner, who led the original ARH-GAP11B research in Dresden, has described the finding less as an answer than as an opening, a demonstration of how much remains genuinely unknown about the ordinary biological mechanics that quietly, gradually built a thinking, questioning species out of raw, unremarkable, previously discarded material. The unexplained, in other words, was never proof of an outside hand reaching in. It is proof of how incomplete the inside story still is, even now, even with every modern tool pointed directly at the problem. Researchers are careful, still, not to overstate what any of this settles. Finding one orphan gene tied to brain expansion does not explain every unexplained fragment sitting elsewhere in the genome, waiting its turn. It only proves that the discarded material, taken as a whole, deserved a far closer look than it originally received, and that the closer look is nowhere near finished. They rebuilt the map of the genome, revised. They rebuilt the definition of junk, revised. They rebuilt the explanation for where a human brain gets its capacity to fold, to reason, to wonder about its own origins. Revised again, revised, and still not finished, and possibly never finished within any single research career. Every person alive carries fragments of an instruction manual that has not finished being translated, and likely never will be inside any single human lifetime, no matter how much funding arrives or how fast the sequencers get. Some of those fragments switch other genes on and off at precisely the right developmental moment, down to the hour. Some fold a smooth, ordinary primate brain into the wrinkled shape that lets it reason, and doubt, and eventually ask where its own code came from in the first place. And some remain, even now, unread. Not because no one has looked, not because the funding was never there, but because looking closely has only revealed how much further there still is to go. What began as a discarded majority of the genome, dismissed for the better part of a century as evolutionary trash swept into a corner, has become one of the last true frontiers inside the human body itself, closer than any distant planet, and in its own quiet way, harder to reach than most of them. The part of human DNA scientists still cannot explain was never junk, it was only unread. And somewhere in a lab, tonight, long after everyone else has gone home, the machine is still running the same query, waiting for a match that has not arrived yet,